presentation is going to be about photosynthesis. My name is Ashley. This is Elijah, Kathleen, and Grace. Okay, so I'm just going to give you a little background information about photosynthesis. Can anyone tell me what photosynthesis is before I explain it? No one? Okay. Okay, so photosynthesis is the process where plants and other autotrophic organisms such as algae can convert light energy, usually from the sun, into chemical energy. And this is how they create food for themselves. So photosynthesis occurs when a plant absorbs light energy from the sun, and then the light energy is captured in the reaction center of a certain pigment, and that pigment is called chlorophyll, and that's located in the chloroplast. So the light energy is captured in the reaction center, and this is when the electrons are excited in the reaction center, and then they produce more energy for the plant. And this occurs with the help of NADPH, which transfers electrons throughout this process, and also ATP, which creates more energy for the process. And the products from this whole process are carbohydrates in the form of glucose and oxygen. So the overall formula for photosynthesis is 6 carbon dioxide, and the carbon dioxide comes from the atmosphere, plus 6 H2O, that's water. That can come from the atmosphere as well as the soil. The plants can pull it up from its roots. And light energy, which comes from the sun, and that inputs energy into the process. And your outputs are glucose, which is a carbohydrate, and also oxygen. And the oxygen in this process comes from the H2O. During the process, H2O is hydrolyzed, and that gives you, it separates the H plus protons, which creates more energy in the photosynthesis process, and that also outputs the oxygen molecules. And that's where you get your oxygen from. Okay, so next I'm going to talk about light absorption in plant pigments. Can I have some actual plants here for you guys? So we have keratinine, which is the orange pigment down here. It's found in orange leaves as well as carrots. And we have chlorophyll, A and B. It's found in your green leaves. And xanthophyll, which is yellow, but it's showing up kind of orange on here, but I have a plant here, and that's in your yellow leaves. If y'all would like to pass it around and look at it. Okay. A marker, yes. No. Okay. Next I'm going to talk about the visible light spectrum. So the visible light spectrum ranges from 400 nanometers to approximately 700 nanometers. And 700 nanometers is your long wavelengths and your low energy. And then 400 nanometers are your short wavelengths and high energy. And also, the visible light spectrum has colors, which is Roy G. Biv, also known as red, orange, yellow, green, blue, indigo, and violet. And red has your lowest energy, and violet has your highest energy. So basically, our photosynthesis experiment was conducted in order to determine exactly how efficiently the plant pigments were absorb light at a certain wavelength.
Okay, and now we're going to get into the experiment part. And first, here's your materials. You first need to have chromatography paper, which is the little white square that we have. It'll absorb the pigments. And then you'll have your capillary tube, which helps with the spinach extract, pulling it up. your plastic wrap, which goes over the beaker, and then some scissors for the extraction. Your spinach extract, you need a 600 milliliter beaker. Well, it can really be any beaker, because it only needs a little bit of solution. But you have 3 test tubes, which will help with the extraction of the pigments, a pencil, and the isopropyl alcohol. Can anybody tell me what the isopropyl alcohol does?
It extracts the pigments.
You have your separation solvent, which helps with the absorption of the chromatography paper. Your spectrophotometer and cuvettes, which will measure the OD with, and staples. So first we have to prepare the sample, and you're going to get your piece of chromatography paper, and 2 centimeters up from the bottom you have to draw a straight line with the pencil. And you also have your spinach extract. And using the capillary tube, you're going to draw up some spinach extract, and you have to draw over the line, the pencil line, about 10 times, and you have to make sure that it dries in between each application. And then, so now the actual separation begins. You have to, first with the chromatography paper, you have to form it into a little cylinder, and you're going to staple it at the top and the bottom, but you have to make sure that it doesn't overlap. And then into your beaker, you have your solvent, your separation solvent. It's only 20 milliliters. And when you put your cylinder in there, you have to make sure that it's barely touching at the bottom. You don't want the separation solvent to touch the line. And then you just let it sit there until it's done ascending, or wait for 15 minutes. And then you'll take the cylinder out and let it dry and remove the staple after extract. And then you have to actually extract the pigment. So when it's done and dried, there's going to be there's going to be a bunch of different bands, but we use 3 of them, and you have to cut each of the bands. And when you have the separate bands, you cut each of those bands into smaller pieces and put them into 3 separate test tubes. And in those test tubes, you're going to have isopropyl alcohol, 3 milliliters of it, which will help extract the pigments. And once they're all extracted, you are going to take your alcohol and the pigment solution in your test tubes, and you're going to put it into cuvettes. And you want to make sure that you don't get any of the paper into the cuvette. And then you're going to have a 4th cuvette, and you're going to put 2/3 full of the isopropyl alcohol, and you're going to use that as a blink when you measure the OD.
Okay, so now I'm going to talk about our results. So as previously mentioned, when we used the spectrophotometer to measure, we measured in 20 nanometer increments all the way from 400 to the beginning of the light spectrum to 720, which is a little past, but just for good measure. So this are our wavelengths, and this is our absorbance readings for all 3 different pigments. So this is the graph of our data. So you can see where the spikes are is where it's absorbing the most for each one. And what we can get from this graph is that chlorophyll A, which is this one, the medium green one, is that it absorbs the most, which is 460 which absorbs the most at 460 nanometers, and it absorbs 0.224. Chlorophyll B absorbs the most at 420 nanometers, and it absorbs 0.232. Carotenoids absorbs the most at 440 nanometers, and that is 0.153. So overall, chlorophyll B absorbs the most because it absorbs 0.232. Chlorophyll A absorbs most efficiently at the highest wavelength, which is 460 nanometers, and that carotenoids absorb the least overall because it only absorbs 0.153. So it absorbs the least light out of all the pigments.
Okay, so, well, why does this matter? Like, why are we recording this? Well, it's because we want to know, like, on what wavelengths the leaves are efficient at absorbing things. So, on this graph, it shows the colors along the wavelength, like along the graph that we made, and we see that it absorbs light in the violet spectrum as well as the red spectrum, but the only area of the graph where none of the pigments absorb is the green spectrum, which is why plants appear green. and that's supposed to be green, but it's kind of black. This is significant because of solar panel research. Can anyone tell me why solar panels are black? Or why they think solar panels will be black? Yeah?
Because it absorbs most of that.
Right. Solar panels are black because black absorbs the most. If we were to do an OD test of like a black pigment, this is the one that doesn't work, it would probably be something like this over all the spectrum because it absorbs most of it. When you see black, you're not seeing a color, you're seeing the absence of color. You can go to the next slide. So, and that begs the question why plants aren't black, but that's really, they've done a lot of research into it actually, and they really don't know why plants are black. They don't know why plants aren't blue or green or orange or red, but because there could be a planet out there where the plants are blue or black. But for now, on this planet, they're green. And so we know that they absorb light in the violet and the red spectrum.